Thank you, everyone. I'm Gerard. I'm the Chief Business Officer at SpliceBio. We are a genetic medicines company, mostly working in ophthalmology, but we've recently generated some exciting data in non-human primates, in the CNS of non-human primates, and that's what I'll be focusing on today. So we have a novel technology in the genetic medicine space that essentially allows us to deliver genes that exceed the packaging capacity of AAV vectors. That's a major limitation in the gene therapy space, where you cannot package the cargo that you like to deliver just because the cargo is too large and the vector is too small. And we get around that by delivering a dual AAV that is super efficient compared to prior approaches. Our lead program, as I said, it is in the clinic. It's a dual AAV in Stargardt disease, first in class gene therapy, completed the phase 1 part of the phase 1/2, and now it's enrolling patients in the efficacy part of the study, dose expansion, randomized and adequately controlled. It's supported by an observational clinical trial that we've been running for some years now, and we have emerging pipeline of programs in ophthalmology and CNS with validation across more than 20 therapeutic targets and validation in the retina and CNS in non-human primates, which I'll show some of it in here, and obviously a robust and strong IP position across platform as well as programs. We've raised over $200 million until now in Series A and Series B financing. We have EQT, Sanofi, and Roche that participated in the Series B, as well as NEA, UCB, Izios, Novartis, Gilde, and Asabis from the Series A. And this is the team that is leading the company. I am the Chief Business Officer and have been since 2016. And this is the main challenge that we are addressing, right? In the genetic medicine space, the limited packaging capacity of these viral vectors that essentially precludes you from doing gene therapy for many of the targets that are actually interesting. There's been several people trying to solve this problem before with other dual AAV technologies. There are mostly 3 of them, right? They rely on recombination at the DNA level, recombination at the RNA level, or recombination/splicing at the protein level, right? And within recombination at the protein level, there is the first generation protein splicing approach using older versions of the technology. And then there is the third generation protein splicing, which is the one that we've been pioneering since we discovered protein splicing in the 90s. And we understood the limitations and challenges of first generation approaches. Now, the way the technology works is that when you have a gene that is too large to be packaged in a single vector, we can split that gene into halves. To each of these halves, we add these sequences encoding for third generation inteins, which are protein domains that catalyze this protein splicing reaction, right? So splicing, but at the protein level. And then we package each of these half transgenes in a separate AAV vector. This dual AAV vector transduces the cell, and then you have expression and translation of each of these halves, and it is at the protein level that these inteins will find one another. They will form a non-covalent protein-protein interaction that will trigger the protein splicing reaction, whereby they will essentially ligate the last amino acid of the first fragment with the first amino acid of the second fragment and reconstitute the full-length protein that you wanted to deliver in the first place. And this protein looks exactly like the native protein. Now, we've deployed this technology across the board, across several therapeutic areas, tissues, and species. And obviously, we are now in humans with our lead program in Stargardt disease. This is how the drug product looks like. So you have 2 AAV vectors. The transgene is split in 2 halves, each of them packaging half of the transgene and the intein. And then when that is delivered at the protein level, the product comes together, and you have, in this case, the ABCA4 full-length protein being made and restoring function in these photoreceptors. I will not go through the data in ophthalmology. I want to jump into the CNS data. But one thing I wanted to show is that we have extensive expression data, so showing that we have high levels of expression, in this case of the ABCA4 RNA and protein in rodents as well as non-human primates. Most importantly, the protein that is being made is trafficked and localized adequately, in this case in the outer segment of photoreceptors. ABCA4 is a protein that localizes very precisely in that region. And last but not least, we have functional data of this protein and topology data proving that the protein looks exactly like the wild-type native ABCA4 protein in humans. Now, this program is in the clinic. We've gone through, we're enrolling across 21 sites globally, mostly in the US and the UK, and we've completed the part A of the phase 1/2 studies, which is the dose escalation, and we've gone through all the DSMB approvals. We've dosed 9 patients, no DLTs, no drug-related SAEs, and now we're enrolling patients in the dose expansion efficacy part of the study, phase 2, up to 57 patients in a randomized and controlled manner for the efficacy data. And now jumping into the CNS data and validation of the technology in the CNS, we have a program in our pipeline in a monogenic CNS indication. And what we've done is prove that the technology is working both in rodents as well as in non-human primates, right? And I'm going to show some data with reporter genes. And what we're doing is we're dosing, in this case, mice intracerebrally, and we're dosing them with a single AAV that delivers the reporter gene, because the reporter gene fits in a single AAV. And then we can split that reporter gene in a dual AAV and deliver that. And we can compare how much protein we get in each case, right? And here you can see how much protein we get with a single AAV in mice and how much protein we get with a dual AAV in mice, right? And you can see that the bands are quite similar and consistent. And then when we dose with each of the AAVs, that's your negative control. And obviously you show no expression of the protein because the 2 fragments are not there for proper reconstitution. And when you quantify the blood, what you see is that we can achieve high efficiency of the system in rodents in this case. Therefore, laying the foundation for you to be able to deploy this technology in genes that are too large in the CNS, right, and that do not fit the AAV vector. We also have epifluorescent data, right, with, in this case, GFP. You have 3 mice here, this with a single AAV, and this is the kind of signal that you can see when you dose these patients ICV, these mice, sorry, not patients, ICV. And when you do the dual AAV using our 3rd generation protein splicing technology, this is the kind of data that you get as well. So comparable efficiency between the single and the dual in mice. And then jumping into non-human primates, we do something similar here. We're injecting intracerebrally, so into the putamen of these macaques. We have the single AAV data with the reporter gene, and this is how the fluorescence looks like with a single AAV delivering the reporter gene. And then when we split that reporter gene with a dual AAV 3rd generation protein splicing technology, this is how the signal looks like. And what you can see is that we have consistent, robust data with a dual AAV system in the brain of non-human primates as well. That is consistent with the data that we've generated in the retina in non-human primates as well as consistent with the mouse data that we've generated across the board. And that's pretty much it. So we have a pipeline of programs in the retina and the CNS. And yeah, happy to take any questions if we have time.
Very quick.